Hello, and welcome to today's webcast. My name is Abigail, and I will be your event specialist today. All lines have been placed on mute to prevent any background noise. Please note that today's webcast is being recorded. During the presentation, we will have a question and answer session. You can submit text questions at any time. Click the green Q&A icon on the lower left-hand corner of your screen, type your question in the open area, and click Submit. If you would like to view the presentation in a full screen view, click the full screen button in the lower right-hand corner of your screen. Press the Escape key on your keyboard to return to your original view. For optimal viewing and participation, please disable your pop-up blockers. And finally, should you need technical assistance, as a best practice, we suggest you first refresh your browser. If that does not resolve the issue, please click on the support option in the upper right-hand corner of your screen for online troubleshooting. It is now my pleasure to turn today's program over to Patrick Lowe, Senior Editor of Biotechniques. The floor is yours.
Welcome to today's webinar titled Flow Cytometry, Emerging Instrumentation and Application Trends, brought to you by Biotechniques and sponsored by ACEA. Hello, my name is Patrick Lowe, and I am a Senior Editor with Biotechniques. In this webinar, our speakers will describe recent developments in flow cytometry technology that have led to the emergence of lower cost and smaller flow cytometry instruments capable of analyzing multiple fluorescent colors. And they will discuss the applications of this new generation of flow cytometers to basic and medical research. Following these presentations, there will be a question and answer session with the speakers, so please feel free to enter any questions into your webinar console throughout the presentations. We are pleased to have Dr. William Telford as our first speaker. Dr. Telford is with the Experimental Transplantation and Immunology branch of the NCI-NIH and is the head of the NCI-NIH Flow Cytometry Core Facility. He will describe recent advances in photonics and how that has enabled the design of low-cost flow cytometers capable of high-dimensional analysis. Bill?
Okay, well, thank you, Patrick. So as the first of these three talks, I'd like to talk about what I think is an exciting area in the area of flow cytometry, namely the application of lower-cost technologies to doing high-dimensional analysis by flow cytometry. Now, this BD Fortessa is typical of what we think of when we look at instruments that can do high-dimensional analysis. High-dimensional being anything more than 8 to 10 color analysis, where you're really looking at many fluorescent parameters simultaneously. This instrument in particular can do 18 colors. It has 6 lasers, so it has considerable excitation capability. And it also has 18 distinct fluorescent detectors. So you can do very high-dimensional analysis on this instrument. However, instruments of this type, while typical, tend to be very expensive. They tend to be very, very large. And what we're starting to see in the field now are smaller, less expensive instruments that are capable of doing much the same type of analysis by flow cytometry. This technological area actually has quite a long history. We've already had a number of small instruments like the BD Accuray, the Guava series of flow cytometers, the more recent HandEM and EMD Millipore Muse instruments that are capable of 1 or 2 color analysis. We also have instruments like the Alera Pima and the BD Presto that are capable of point-of-care analysis, where you're also doing only 1 and 2 color. But these instruments tend to be very simple and capable of only a small number of parameters. More recently, though, we have seen the development of some instruments that are capable of more than this. The originally DACO Cytomation and then Beckman Coulter Cyan on the top up here was a 9-color flow cytometer developed about 6 or 7 years ago, a very capable instrument at the time with 3 lasers that could take advantage not only of the traditional blue, green, and red excited fluorochromes, but also some of the newer violet ones at all. The Partec Sysmex instruments below were also capable of doing more than 10 colors using multiple lasers. However, these instruments still tended to be relatively expensive. They were not much cheaper than the more traditional larger instruments. What we see more recently, though, are smaller frame instruments where the cost is starting to come down. The Miltenyi Biotech is on the left. This is a 10-color flow cytometer that uses 3 lasers, a very small footprint, very easy to maintain. The Stratadyme instruments are on the right. These are also small cytometers that can be equipped with 4 or more lasers and do up to 18 fluorescent parameters. So we now have a lot of instruments like this on the market that are capable of this level analysis. the Miltenyi instruments, the ACEA Biosciences NovaSite, the Thermo Fisher Attune Next, the Guava series manufactured by EMD Millifor, the more recent Beckman Coulter Cytoflex, and the BT Biosciences Celesta are actually only a sampling of the instrumentation that we have out there. Not only are these instruments capable of high-dimensional analysis, but they also rely on new technologies that allow flow cytometry to be done better, but also at a lower cost. So what are we considering a high-dimensional cytometer to be? We want to have multiple lasers, at least 3, the blue, green, red, and violet, and additional laser options are, of course, very desirable. 10 or fluorescent parameters at least, and this can be more than that. We want to be able to modify our optics for special applications, as we're able to do on the larger machines. We'd like to be able to have flexible sampling handling options, tubes, or microplates. We, of course, want the software to be user-friendly and to do things like full matrix compensation. In short, we want these systems to do everything that the traditional machines can do, but we want it to be smaller and hopefully cheaper. And by employing newer technologies, this is now very possible. Here are some of the modern platforms that are available that I just listed. The Guava EasySight series, which ranges anywhere from 3 or 4 colors up to more than 10, using 3 or 4 lasers. The Beckman Coulter Cytoflex, the Attune Next. I'd like to concentrate, though, on one platform that is representative of these technologies, the ACEA NovaSite. This is a recently developed small-scale flow cytometer, very small footprint. It has a 488, a red, and a violet laser on it, and it can detect up to 13 fluorescent parameters simultaneously. The ACEA NovaSite takes advantage of an interesting mechanism for delivering sample to the instrument. Most flow cytometers are hydrostatic positive pressure systems. They use pressurized sheath tanks to deliver sheath to the instrument, and then a pressurized system also to deliver sample. And almost all flow cytometers today rely on hydrodynamic focusing, where we inject the sample into an outer cylindrical sheath flow that focuses the sample into the center of a stream or a cuvette. Whenever you're filling up the sheath tank, what you're doing is providing sheath buffer that will focus that sample into the center of the stream. This is what a typical flow cell looks like. This is off of a BD fax caliper. You can see the tiny little quartz flow cell contained in the center of the overall flow cell construct on the right, where the sample is delivered through a SIP or a sample tube in the bottom. It's merged with the sheath stream, and then it's focused in that quartz cuvette. The lasers are focused on that cuvette, and the signal is then transmitted back to the instrument optics, a very traditional design. And as I mentioned, most instruments use positive air pressure. They use a pressurized sheath tank and also a pressurized sample tube using air pumps to deliver both sheath and sample to the flow cell, and ultimately to allow the cells in that stream to be analyzed. However, there are other technologies out there that can deliver both sheath and sample. Syringe pumps have been used on a number of flow cytometry systems for delivering both sample and sheath, or often just sample. We actually inject the sample into the sheath stream. This is a nice system because the delivery is very uniform. We know the volume. We can calculate absolute counts. Some newer generations of instruments are also using peristaltic pumps to deliver both sheath buffer and inject the sample. And since the volume is also known on these pumps, we can also derive absolute counts as a consequence of the sample delivery system. Now, the way the NovaSite works is it uses a sample syringe pump delivery system located on the lower right-hand corner of this diagram. This is the instrument with the cover off. So this particular instrument will deliver absolute counts and can also deliver multiple syringe loads of sample to the instrument without interruption. So even though the syringe may be small in volume, you're able to deliver large numbers of samples to the instrument. Now, multiple samples and many detectors are now normal on a flow cytometer. The earliest instruments had only 1 or 2 lasers. Advanced instruments can have 7 or more lasers to take advantage of the hundreds of different fluorescent probes that are out there. Typically, though, instruments now take advantage of 3 laser wavelengths: the blue 488, the red 633 to 642, and a violet 405. And even with just these 3 laser sources, you can analyze up to 16 fluorescent probes simultaneously. These lasers have also gotten very small. The original lasers on benchtop machines were air-cooled argon lasers. The size has now been reduced to a laser that you can hold in the palm of your hand. This is a Fortran Stratus Lite 488 nanometer laser, a 20 milliwatt module that I'm literally holding in my hand down there. The lasers have become very small. The cost is also starting to come down. The NovaSight takes advantage of this. It has 3 coherent lasers, very small units that it uses for excitation of fluorescent probes, a blue, a red, and a violet. The laser beams are spatially separated like they are on traditional instruments. The NovaSight also takes advantage of PMT sharing, which is another interesting development in lowering the cost of cytometers. Here you can see the optical bench of the instrument, the filters and dichroics that we need to both separate and filter out the fluorescent signals are present. But even though it's a 13-color machine, it only has 6 PMTs in it, plus 1 for side scatter. What it is able to do is reuse the PMTs for fluorescent probes that have different excitation but similar emission properties. So this is what the bench looks like over here. This is the plane for 488 blue laser excitation and analysis, where we're analyzing FITC, PE, PE Psi 5, PE Psi 7. So the cell passes through the first laser, and the detectors become active for the particular fluorochromes that correspond to the filters in front of the detectors. Once the cell moves to the next position in the red laser, the same PMTs are used, but the signals go to different electronics. So the APC, for example, is now able to be detected in the PE Psi 5 detector, and APC Psi 7 is detected in the PE Psi 7 detector. The same with the violet. We're able to take advantage of all 6 PMTs to detect all the brilliant violet dyes, and again, we're using the same detectors off of a different laser. This lowers the cost of the instrument considerably. PMTs are an expensive component in a flow cytometer, and it makes the instrument less expensive without reducing its functionality. These instruments are sensitive. We are able to verify this using calibration particles, Spherotech rainbow beads, MESF beads for FITC, for PE, for different fluorescent probes. And what we have found on all of these platforms is that the sensitivity is comparable to what we get off of larger traditional instrumentation. Here we're looking at Bang's FITC MESF microparticles on the right-hand column, and we're able to distinguish the dimmest population almost to the same degree on the NovaSight as we are on a BD LSR for TESA. So we don't lose sensitivity by going to these lower cost platforms. The ACEA system and other cytometers as well also take advantage of detectors and electronics with very wide dynamic ranges. The ACEA has fixed PMT voltages. You do not adjust the PMT voltages as we're accustomed to doing on older systems. You collect a very wide dynamic range, and then you zoom in on the area that you want to see and analyze your data. I think this is a wonderful feature. A loss of sensitivity is very often the result of turning up a PMT too high or too low. We also tend to complicate our compensation values by being outside the best possible settings for a PMT. If you have factory set PMTs, you don't have to worry about this. You know that the PMT is at its most sensitive and its most linear point, and then you can zoom in on the area that you want. So we have done that here, both with forward scatter on the upper pair of dot plots and on histograms for some rainbow beads on the lower end dot plots. I think all cytometers should be like this, and I think in 10 years from now, we are going to either be using different detectors from TMTs or we'll be using locked PMTs where we no longer adjust the values. The newer instruments also are able to take advantage of the full array of fluorescent probes that are out there. We're very fortunate these days. We have a wonderful array of fluorescent probes available to us. The brilliant violet, the brilliant ultraviolet, and brilliant blue probes from BD Sirigen that have greatly expanded our ability to do high-dimensional flow cytometry. Currently, there are 6 or 7 different brilliant violet dyes available, BV421 up to BV785. We also have the brilliant ultraviolets and the brilliant blues as well. This allows us to add a large number of parameters to our high-dimensional panels. And many of the newer instruments take full advantage of this capability. The Novasight has 6 detectors that can be tasked to measure BV421 in the blue all the way up to BV787 in the near infrared, and we're able to see all of them. So the Novasight and other platforms like it have been designed to take advantage of all the various fluorescent probes that we now have available to us. So I think that small high-dimensional cytometers are dramatically changing flow cytometry. The cost has come down. Cytometry is becoming cheaper, but not necessarily with losing capability. We're able to do complex multi-color experiments using smaller, less expensive instrumentation. These instruments are often easier to service, and it's often depot service where you pack the machine up, ship it back to the company, and they send you a loaner while they're working on it. So downtime can be minimal, and service can be more available to areas that are more remote where service engineer coverage is often not as assured. These instruments often offer specialized laser wavelengths, the blue, the green, the yellow, the ultraviolet, previously available only on expensive instruments. These instruments can be set up and maintained in more remote location. We're seeing a lot of new technologies being worked into these instruments as well. Avalanche photodiodes, APDs, are starting to work their way into flow cytometers. These detectors are very sensitive, yet also very inexpensive. You're also starting to see this instrumentation be approved for clinical use in other countries. The Novasight and the Cytoflex from ACEA and Beckman Coulter are both approved for clinical use in China, for example. So we're going to start seeing this technology also make its way over into the clinic as well. So I will end my talk there. Thank you.
Thank you, Bill, for your presentation. Our next speaker is Dr. Paul Wallace. Dr. Wallace is Professor of Oncology as well as the Director of the Flow and Image Cytometry Department at the Roswell Park Cancer Institute. Dr. Wallace will discuss his research using multicolor flow cytometry and fluorescent cell tracking dyes to analyze the precursor frequencies of subsets of T cells and the proliferation. Paul?
Thank you very much, Patrick. Before I get going, I'd just like to acknowledge some of my collaborators on these projects, and that's Catherine Muirhead, who's been a longtime collaborator going back several decades, Kylie Price at the Mulligan in New Zealand, and Joe Tario and Katiang Sa at Roswell Park, who really have sort of worked with us through the years on these tracking dyes. So to begin, what I'm going to be talking about is sort of an introduction to what the dye dilution technique is and some of the key assumptions. We'll talk about differences between membrane and protein tracking dyes, just briefly discuss some of the analysis strategies, and then look at some of the key differences between the different dye types. As a way of background, the idea of the dye dilution assay is that you would stain up some cells, very often lymphocytes, with a nice bright dye, and then stimulate them in some way. And as they divide, the daughter cells that are produced will have half the fluorescence intensity of the parental cell, and of course they'll double. So if in generation 0 we have 10 cells, for example, we will stain them up, and if one of those cells is going to go on and divide, say respond to a mitogen or to an antigen, we'll now have, that'll be 10%. But then as it goes forward, in the first generation, now that fluorescence intensity of the responding cell will be half, or 50%, and the percentage of responders will be 18. And as we go through further down to, say, the 5th generation, at this point, 78% of the cells are related back to that initial responder, and the fluorescence intensity has decreased to about 3%. So in this way, we can really use this technique to measure a lot of characteristics about the cell. So in this, there's some basic key assumptions that we basically take for granted, which as we go through this may or may not be true. The first idea is that the proportionality between an increase in cell number and a decrease in dye fluorescence is the same at each division. So essentially what that means is that the number of cells are going to double, and the fluorescence intensity is going to decrease by half with each consecutive generation. However, what we'll find is that really isn't always the case, that decreases in fluorescence intensity due to cell division can be distinguished from other cases, but the decrease may not be due just to the decrease as a consequence of division. In fact, the reduced fluorescence intensity can be to metabolism, export of proteins or other biological processes that are occurring within the cell that may cause the dye to be degraded. There's a whole variety of commercially available proliferation dyes, and these are generally divided up into protein labeling dyes and membrane labeling dyes. They come in a wide variety of colors. For example, we have the cell trace violet, which is excited off of the 405. That's a protein labeling dye. Sort of the prototype is CFSE, and that's excited off the 488. And then we have ones such as the E4 670, which is excited off of the 640 line. In the protein labeling dyes, there's the same variety. Cellview lavender off of the 405. Really the prototype for this is PKH26, which emits in the red, and it's excited off of the 488. And then Cellview claret off of the 640, and even off in the infrared, the Cellview NUR 815s, which are excited off of the 780 laser. There's a wide variety of applications and notes, and all these have been published on. The idea is that for cells labeled with the tracking dyes, there are basically two different types, as I mentioned in the previous slide. The first is the protein labeling, and the way this works is really best outlined with CFSE, which is more or less the one that's been around for the longest, and it's really become the prototype. When you stain up the cells, it's a very rapid uptake of the dye inside the cells. As it's basically a non-fluorescent dye. Then when it enters the cell, intracellular esterases will cleave the dye. It'll attach to a protein and it'll become fluorescent. At that point, it's now polar and is no longer to escape from the cell. So the cell inside the cell is now a fluorescent color. These are covalent bonds, so they're stable and they remain there for a very long period of time. Disadvantages are that because you're staining up proteins, the dyes themselves can actually affect protein function. And we've certainly seen examples where we've put too much dye in and then the cells aren't able to proliferate for several days. The other thing that we'll spend some time thinking about this is that there's often a very high loss of dye, particularly at between day 0 and day 1, that probably have something to do with the metabolism of the dye or export. In fact, it's really not clear to any of us exactly why you see this rapid turnover. The other type of dye are the membrane protein tracking dyes, and this is best exemplified by PKH26. And these basically have these long tails, these carbon tails that will intercalate into the membrane. It's not covalent, but it's basically the tails will go into the membrane, and thus the cells become fluorescent. The intensity the fluorescent staining is very rapid, really occurring probably in less than a minute. And because of the length of the tails, there's very little or if any loss of the dye from the cell. The disadvantages, probably the biggest one, is that the fact that it's a little bit more complicated than doing the technique is a little bit more complicated than using the protein dyes. You can certainly put too much dye in and lyse the cell, and as I mentioned, the dye is not covalently bonded. It's actually maintained through hydrophobic interactions. So we have these two different dye types. This slide illustrates the concept of looking at cell division. It was some work that was done with PKH26. And the way this experiment in the upper left was carried out was some beads were placed into a culture with some stained cells. And then on day 1 through day 5, a sample was extracted from the flask, and a constant number of beads were enumerated. That's the box that you can see in the very upper left-hand corner of the inserted histogram. So the same number of beads were counted, but as you can see in the actual histogram itself, at day 0 we count a few cells, but then with each successive day, counting essentially the same volume of cells, the same volume of liquid, we're counting more and more cells. And the fluorescence intensity is decreasing. On the right-hand side, what you can see is that we were plotting the MFI, or the fluorescence intensity, on the x-axis at time 0, divided by the fluorescence intensity at a later time, for example, day 1, day 2, day 3. And so what this is going to do is it's going to tell us the decrease in MFI. So with each division, the theoretical decrease would be 50%. On the y-axis, we're looking at count, and it's essentially the reciprocal. We're taking the count at a subsequent day, day 1, day 2, day 3, day 4, and dividing that by the initial count. So if the culture doubles, then we would expect that number to double as well. And what you can see here is basically whether we look at the decrease in MFI in green or a calculated metric called the proliferation index, that we have a slope of 0.98 and basically a pretty good correlation. So if we go way out to to where the count is at basically 30-fold, we're getting a decrease in fluorescence intensity of about 25-fold. So a fairly good relationship there. In terms of the way you would analyze this, there's a variety of ways. This is an experiment that was done at Roswell by Fong Quan, and what Fong did was to extract CD25 positive and negative cell fractions from CD4 T cells, and then he stimulated them with anti-CD3 and anti-CD28. And in this case, he cultured them for 7 days. So these were stained up with CFSE, and you can see in the upper left-hand plot A, or graph figure A, that the cells start off with at the generation 0, the cells are brightly stained, and then as they divide, the fluorescence intensity decreases. We look in and compare that to the fraction, the CD25 positive fraction, which is probably the T regulatory cells. We get the same type of thing where we're seeing division, but there's not quite as much division as in the CD25 negative fraction. And this can be roughly quantified over on the right-hand side by just dropping anchors down around the actual population that's proliferated, and we can refer to that as the proliferative fraction, which with the 25 negative fraction is 83%, and with the 25 positive fraction is 62%. So in fact, by both of those measures, we can see that the number of cells that went on to divide is lower in the 25 positive fraction, which probably, as I said, represents the T regulatory cells. A more elegant way of analyzing this data is to use a curve fitting software, and these are available from a variety of vendors. I've got here FlowJo, ModFit, and FCS Express, and basically the different plots that you would get from them. But the idea is that you can fit the different peaks that you see in the the proliferating cells, and you come up with a number of events or number of cells in each of the generations. So looking at the FlowJo in the upper left, at generation 0 we have 329 cells, at generation 1, 1,501 cells, etc. And we can do that with all the software. And the reason that's of value is because of what I would refer to as my jelly bean slides. This is best illustrated here, where we can actually begin to calculate out what is known as the precursor frequency or the proliferation index. And so the idea here is that with the precursor frequency, for example, this is essentially the number of cells in the parental generation, the number of cells in the original starting culture that went on to divide. And so if we look at the, on the bottom there, if we look at basically the colored jelly beans, you can see that in generation 6, we had 64 events, or 64 cells. And in generation 5, 160, and in generation 4, 48. And then in the ones that were undivided, we'll say 100. And this is supposed to represent sort of the histogram that we have up above, which were cells that were stimulated with a tetanus toxoid in patients that had, or in individuals who had been vaccinated with tetanus. So what we can do is, with this snapshot, we can then go back and calculate what was in the parental generation. So if in generation 6, we had 64 events, then when they were in generation 5, it would have been 32. In generation 4, it would have been 16. And we can go all the way back to the parental, and there you can see that one red event that in generation 1, that would have been one event or one cell that went on to divide and become 64 in generation 6. And so we can do that with all the generations and basically come up with the jelly bean plot that we have in the lower right-hand corner, where there are 110 events and of those, we basically have 10 events that went on to proliferate, and that would be a precursor frequency of 9.1%. That's the equation in the upper left. So that's an advantage to know that, because that's essentially the number of events that were responsive to a particular antigen, and that's important if we were to do like a vaccine study. The other thing we can calculate out is the proliferation index. In this case, it's the total number of events that you see in the final day 7 culture divided by what the theoretical was in the when you start off with. And in this case, that's 376 divided by 110, which is essentially a 3.4-fold increase in the number of cells while they were in culture. And that's essentially equivalent to like a stimulation index or certainly a fold expansion. So I will caution, this is just a quick slide to indicate that the precursor frequency, for example, in ModFit is called, or in ModFit what we just described as a precursor frequency is called precursor frequency, but in FCS Express and FlowJo it's called percent divided. The proliferation index is what the term that's used in ModFit and FCS Express, but the expansion index is what that term is used in FlowJo. So there's differences between software as to what is being called, what is each of these terms, or what each of these calculations are. Mario Roeder has a great article that appeared in Cytometry A that basically addresses much of this, but that's just a caveat and a word of caution. So here's an example of where we've used the precursor frequency to follow influenza vaccinations in, I think it was 14 individuals, or I guess 9 individuals. And in this case, they were immunized with we took a peripheral blood sample on day 0. They were then immunized with the influenza vaccine, and then 13 days later, cells were calculated, the blood was frozen down, and then we thawed at a later date both of the pre and the post vaccination samples and calculate out precursor frequencies. This was done, as you can see in the figure, or in the bar plot, for both CD4 and CD8 cells. And in general, we saw an increase in precursor frequencies in all the individuals. It was highly significant for CD4s and and not quite significant for the CD8s, with most of the individuals showing a response. We've also done a similar type of experiment where patients with a colon cancer were given a vaccine that was essentially a lysate of their tumor that was mixed with autologous dendritic cells that we had cultured up, and we did precursor frequencies before and after the vaccine. So the patient blood was taken before the vaccine, their tumor was harvested, mixed with the tumor lysate, and then they were vaccinated, and then at various time points subsequently we did precursor frequencies. And what you can see on the right-hand side is basically that individuals that were responders, and we had 6 of these, responders being individuals that showed a increase in precursor frequency, had a much higher recurrence-free survival than individuals where the precursor frequency remained unchanged. And so it really, at least in this example, measuring the precursor frequency by these techniques really predicted the response to the vaccine. So a question comes up in all of this: do membrane and protein dyes perform the same when it comes to measuring the proliferation of cells? And so basically for this, we've done a number of experiments, but really used the NovaCyte here to begin to ask questions about what were the differences between the protein dyes and the membrane dyes. So in this figure, on the left-hand side, we have protein-reactive dyes. On the right-hand side, we have membrane. And we're basically just looking at that same plot that I described earlier. We're looking at the fold increase in count on the x-axis and the basically the decrease, or the fold decrease in fluorescence intensity on the y-axis. And for the protein dyes, one thing that is very obvious with the green lines and symbols there is that there's a big difference between CFSE and the other ones. And that's held up for a long period of time. So that there's actually a much bigger decrease in fluorescence intensity than you would predict with the CFSE dyes. And if you look at that curve a little closer, you'll see that in essentially the first day, that decrease is at a much higher rate than the decrease that you see at subsequent days. That's true if you look closely at all the other protein dyes, that there's a different kinetics to the loss of dye in the early stages than there is in the subsequent stages. That's different if we go to the right-hand side, where we have the one green line. You can see that that's coming in from CFSE versus the membrane labeling dyes, which pretty much track the theoretical. That's the black line that's going through the plot. So that the protein dyes, we're seeing less of this unexplained fluorescence loss than we do with the protein-reactive dyes. So we, taking advantage of the NovaCyte, set up an experiment where we co-labeled the same cells with a protein dye and a membrane dye. And basically and then we also did absolute number counts on the NovaCyte because it knows the volume that it's taking. It can then give you the number of cells per microliter. So we stained the cells up with the tracking dye. In this case, we used cell trace violet. Then we incubated those for 20 minutes, did some washing, then we stained them up with the PKH26 or the claret dye, did a quick incubation, stopped, washed, and cultured them for 6 days. And the way that experiment was set up, as shown here on the right, was that we had basically a checkerboard design where we stained them up with 20 micromolar claret and 20 micromolar cell trace violet, or 10 micromolar or 5 micromolar. The idea is that we stain them up at different concentrations of both the claret and the cell trace violet, including one concentration that was without the cell trace violet or without the claret. And we did the same type of thing with the PKH26. Here is basically the gating strategy that was used. We're basically using forward and side scatter to define, and these are U937 cells. We gated out dead cells in the upper right with propidium iodide, and then you can see the fluorescence intensity of both the cell trace violet and the claret. Looking at just cell growth, this is days in culture, and looking at the expansion, it's a log scale on the y-axis, and you can see that these are essentially in log growth. On day 5, you can note, particularly in the middle one there, that there's a slight bump, and that's actually when the culture was changed. It was basically refreshed because the media was getting depleted. So we're in logarithmic growth. This is the data coming off of the Fortessa, and we're going through from day 1 or day 0 to day 6. You can see clearly the decrease in fluorescent intensity for the cell trace violet, the PKH26, and the claret. These are all at the higher concentrations, and one thing I'll point out is that it's off scale at day 0 and actually at day 1 for the cell trace violet as well as for the claret. And this is data coming off of Fortessa. Here's the data off of the Nova site, and you can see it's the same data, same exact samples, that because of the increased dynamic range of the instrument, with I think it's over 7 decades that we're able to get all that data on scale. So here's the data looking at count on the x-axis, increase in count versus the MFI on the y-axis. If we take a look at the plot in the upper left-hand corner, the first thing you can see is that the blue line, that's cell trace violet only, there's no PKH26, that is somewhat retarded. We would expect this would have expanded about 25 or so fold, and in fact we're not quite seeing that much. If we look at the cell count only, and we're looking at just the blue line, the increase in cell number is about 20-fold, whereas the decrease in fluorescence intensity of that blue line on the y-axis there is about 150. So we're seeing a lot more dye loss than we are increase in cell number. And that was true for, it's really true for both the blue line, the cell trace only, as well as the red line, which is cell trace violet, also staying with PKH26, and for the green one, which is cell trace violet, and a PKH26. The one exception is the purple line there. In that case, we've got just the PKH26 staining. If we go, the same is essentially true if we look at the next one over to the right, but because this is now looking at day 1 as our denominator or numerator as opposed to day 0, that change isn't nearly as much, and that's because of this rapid efflux between day 0 and day 1. If we go down to the very bottom, lower right, what you can see here is essentially we've pretty much got everything normalized. We're seeing that with both the green, the PKH26 staining, and the even whether it's stained with the cell trace violet at a low concentration or not, we're getting good reproducibility. It's at about 25 on both the x-axis for count and 25 on the y-axis for decrease in fluorescence intensity. Whereas if we're measuring the cell trace violet, that decrease is slightly more. So that's a complicated slide, but I think it nicely illustrates basically using the instrument to look at changes in fluorescence intensity on cells that are stained, simultaneously stained with different types of dyes, and incorporating into it a cell count. We've gone on and we've done this a number of times. This is now looking at the cell trace violet data with the claret, and essentially if you look at the upper left-hand corner, it's essentially the same interpretation as we had for the earlier slide that I went through in much greater detail. So this is some work that has been done by Matera on the left-hand side, where they've taken iGrove slides, or cell lines, and you can see exactly what I'm talking about here now with the CFSC, where the blue is basically at basically time 0, and then as you go to the cyan and the red, we're increasing by a day. So there's a big drop off between day 0 and day 1, and then a relatively consistent drop off from day 1 to 2 and day 2 to 3. The fact is that we can actually model that. So we can actually come up with models for this by staining up a culture and then not stimulating it and just watching at what or not allowing it to proliferate, and just watching what the efflux is going to be. And that's what's being plotted down here on in the lower left-hand corner, where with the triangles, this is fitting of the CFSE data of these cells without trying to model the efflux over time. And the diamonds are basically with that efflux calculation. So we can actually model this using different conditions and running a control that does not go on to proliferate. So I'd like to conclude by going over some of the things we've talked about here, and that is that cell tracking dyes, the cell trace and PKH, etc., can be used to monitor proliferation, but there are some caveats to it, and that's basically this efflux that I'm talking about. Fluorescence is approximately or inversely proportional to the number of cell divisions in continuous culture. But you have to be aware of the rapid efflux as well as the slow efflux in doing your calculations. Multiple data analysis techniques exist. I showed you where we just looked at the proliferative fraction, but we can really do some modeling using a least squares analysis. Showed you some data looking at precursor frequencies and how these can be used to monitor the effects of vaccinations. And the last point here, I actually didn't show you any data, but we've certainly done quite a bit of work with it, showing that by looking at these precursor frequencies and looking at the proliferation index, we can use these as surrogates for spot assays and tetramer assays, combine it with a multicolor approach, and look at difference between those responses. They basically correlate very well, although what we find is not every cell that stains with the tetramer also then goes on to proliferate. So with that, I thank you very much and turn it back to you, Patrick.
Thank you, Paul, for your talk. Our next speaker is Dr. Christopher Groves. Our next speaker is Christopher Groves, a senior manager at MedImmune, Inc. He will discuss how recent developments and improvements in flow cytometry workflows can enable and deliver breakthroughs in medicine. Chris?
Thank you very much. Before I begin, I'd like to thank Biotechnique and ACEA Biosciences for their invitation to speak today, and especially I'd like to thank Dr. Patrick Lowe and Renee Nunez. I'm honored also to speak alongside two highly respected and regarded cytometry experts, Dr. Paul Wallace and Bill Salford. The title of my talk today is Enabling Disruptive Science Through 21st Century Technology. What I'll really describe is our effort and vision to evolve cytometry capabilities to deliver innovative medicines. MedImmune is over a 25-year-old international biotechnology company. We were purchased by AstraZeneca, a global pharmaceutical company, in 2007 and merged with Cambridge Antibody Technology. Our three research facilities are in Gaithersburg, Maryland, Cambridge, United Kingdom, and Mountain View, California. Our disease area focuses are oncology, infectious diseases and vaccines, respiratory inflammation and autoimmunity, and that's the team that we exist in, neuroscience, pain, and cardiovascular and metabolic diseases. Therapies or therapeutic platforms are monoclonal antibodies, non-antibody scaffolds, and recombinant proteins. As such, we are the worldwide biologics business unit for AstraZeneca. AstraZeneca, in turn, has the same disease area focuses, but generates solely small molecules or compounds as drugs. So together, we generate both small molecules and large molecules as medicine. As such, utilizing monoclonal antibodies to as drugs, we believe that flow cytometry is a key tool for immunology and a keystone technology for our research, development, and manufacturing efforts at MedImmune. We have over 175 users at our site and over 15 cytometers and 5 cell sorters. We provide core support. We are a core facility and provide training and support to our users. We identify, test, implement novel technologies and methods support and collaborate with research as well as development teams to aid in drug development efforts. In addition, we advocate for the advancement of the art of cytometry throughout the organization and work with site leaders across the globe to promote harmonization of capabilities, foster collaboration, and enhance operations across sites at MedImmune as well as AstraZeneca. Our challenges for flow cytometry are that flow cytometry is incredibly complicated. The technology advances rapidly. New reagents and assays and methods appear seemingly weekly. There are a plethora of instrumentation, as described earlier by Bill, from very simple to incredibly complex, all of which seem to be uniquely configured with different light sources and detector options. The data complexity generated by these systems often requires interpretation and sometimes troubleshooting, which can be challenging. Now, our users, and I think many in the field, seem to believe that there is an art to produce consistently high-quality data. And this may cause some to be afraid to use the technology or to overthink their acquisition and analysis using cytometry. So to enable access and to improve the use of the data and improve the quality of the data that we generate, we believe that we need to try to make cytometry easy. Our idea for this, or our vision for this, is through the use of standardized protocols and methods, incorporation and use of easy-to-use instrumentation to provide on-demand training, and we are learning to embrace automation and automated methods to improve our use of cytometry. In pharmaceutical research, there are constant pressures to improve efficiency and reduce costs. We have devised a roadmap to transform our research to reduce time and cost of research activities. Our simplification efforts, we believe, have provided a productivity gain. Our current state is that our core staff struggle to keep up with the growing demand for researcher access and support, and the data quality generated from our facilities seems dependent on the user skill or experience. We believe that through process improvement and the utilization of standard methods such as omics published in cytometry and easy-to-use flow platforms will allow for integrated research efforts and shared research efforts across our large organization. Furthermore, the future state that we're trying to achieve would allow for the integration of automated processes, dramatically reducing our costs, and FTE, which is full-time employee time, which can improve the quality of our data. We believe that these automated methods represent the future of easy-to-use flow cytometry. The efficient and precise automated processing that saves time, reduced error and variability, and saves costs. We've used off-the-shelf technologies such as the Hamilton Star Fluid Handling System and the high-res biosolutions automation platforms, and worked with a number of flow cytometry platforms and generated data showing a dramatic reduction in average CV or variability compared to manual methods. A critical component to this effort is flow cytometers, which are fit for the purpose of integration into automation platforms, those that are small but highly capable. The NovaCyte from ACEA Bioscience is one of those types of flow cytometers. It is automation ready and easily integrated into the automation systems I'm showing you. In addition, its fixed PMTs offer an advantage to driverless data acquisition. It's our hope that through the adoption of these processes that our scientists can now focus their efforts on the meaning of the results and less on the variables and data outliers. We believe that flow cytometry data is a key component of integrated informatics and can lead to improvements in data mining and sharing across our organization. The reduction in time and cost for both research and clinical application will be something that we hope to utilize to reduce costs of drug development. So what are the other benefits of making flow cytometry easy? Really, to transform the science, we believe that we need new methods to deliver new medicine. Flow cytometry has been a critical capability for our manufacturing and drug development efforts, highlighted by our use of monoclonal antibodies. Our parent company's use of small molecules, or development of small molecules, have been what we have traditionally used to call medicine. However, cell therapy is a new method of treatment that offers potentially curative therapies. From the use of monoclonal antibodies to target cells, cells are not the target, but cells are actually the new medicine. So what we want to do, and our aim and our vision, is to evolve our capability to deliver and support cells as products through the use of GMP analysis and cell sorting, automated processing, and integrated informatics. We aim to deliver curative solutions to patients. Thank you very much for your time. I would like to thank my flow cytometry core team and management, the automation team that we've collaborated with, and ACEA Biosciences for their support.
Great. Thank you, Chris, for your presentation.
Thank you.
We have now reached the end of our presentation. Thank you to all three of our speakers for their talks and to ACEA for their sponsorship of this webinar. We now have some time for our presenters to answer and discuss selected questions from the audience. Hi, our first question is for Bill. Bill, ACEA looks like it has nice sensitivity at the first detectors off of each laser. How does the sensitivity hold up for the detectors further out of the path?
Regarding the ACEA Nova site, the instrument shares detectors between laser paths. So when the cell passes through the first 488 laser, the PMTs are activated and collect FIT-C and PE in the tandems. Then when the cell proceeds to the second red laser, those PMTs that were used for PE Psi5 and PE Psi7 are used for APC and APC Psi7, and then the cell proceeds to the violet laser where the brilliant violet dyes are detected. We really see no difference in sensitivity between that instrument and any of our other PMT-equipped instruments. So this sort of unique approach to PMT sharing and lowering the cost of the instrument does not seem to affect sensitivity at all. We're able to resolve things like the bangs, FIT-C, PE, and MESF particles. They're very low fluorescence and represent only thousands of molecules of fluorescein or PE per bead, and we're able to resolve them without any difficulty.
Great, thank you. Our next question is also for you. Currently, cytometers require CS plus T setups to allow voltage optimization to make results as standardized as possible around the fluctuations in laser and PMT efficiency. How will this be done with factory-fixed PMT voltages?
With factory-fixed PMT voltages, I think one of the important elements of CS&T, which is to determine the best gain setting for both linearity and sensitivity, is not really necessary. So you don't need to, at least as a user, you shouldn't have to perform all of the CS&T functions. You'll still need to do quality control and run some sort of particle. And all cytometer platforms, whether it's the NovaCyte or the Cytoflex or the Miltenyi Max Quant or any others, have a routine for doing this. But doing CS&T and determining the point of maximum PMT sensitivity and linearity should have already been done for you using a factory-fixed PMT. And I think you're going to start finding more instruments like this. The BT Accuray does it, and you're going to start seeing other systems as well that take advantage of this.
Great, thank you. Paul, our next question is for you. Can you recommend good membrane labeling dye that will uniformly label the membrane of cells? PKH26 gives very non-uniform staining of cells in culture and a patchy pattern.
Yeah, that's an interesting question. A lot of people do have problems with the lipophilic dyes. the membrane labeling dyes in the sense that they can be very difficult to work with. They're not as easy to work with as the protein labeling dyes, and so typically it's a techniques thing. And what I can say is that there's actually, I guess I could point people to a Jove video. If you were to put into Jove PKH26, there's a video that really details the procedure for doing that. But in essence, we have a population of cells in one tube that's diluted in PKH26, and another population or another tube with the PKH26 in it. Excuse me, let me start that over again. We have one population of cells that's diluted in diluent C. That's what I meant, is in diluent C. And we have another tube where the dye, the PKH26, is diluted in diluent C. And the trick is to combine those tubes relatively rapidly. And by rapidly, I mean as fast as you possibly can, and not to slowly add them or to slowly mix it, because the staining is virtually instantaneous. So I think really that the dyes do work fine, but they're more difficult to work with than many of or than all of the protein labeling dyes.
Great, thanks. Our next question is for Bill. How does the reuse PMT technology compare to other flow cytometers in respect to electronic aborts? If I understand the approach correctly, event B must not enter the first laser before object A passed by the last laser. Does that matter, or is the time needed to generate a baseline longer than the time it takes to pass all three lasers anyway?
It shouldn't matter, any more than it matters with other digital systems, because right now, in the old days, when peak and hold was used, if you had more than one laser, or if you were sorting, and the cell passed through the laser beam and then was sorted, you had a period where you could not do any other analysis until the cell had cleared the system, which tended to cause high abort rates and very high flow rates. With the digital systems, they can process cells much faster. So it shouldn't really be any more of a problem than any other digital systems now, where you can be processing many events simultaneously, and a cell does not have to clear all of the beam intercepts before another cell can be analyzed. I'm not absolutely 100% sure of this, but I do think that you shouldn't see an issue here any more than you would in, say, a more advanced BD system where you have 5 or 6 laser intercepts. You can analyze more than one cell simultaneously, and that significantly reduces the number of aborts.
Great, thank you. Paul, our next question is for you. What is the best way to assess whether you have added too much protein labeling dye and affected cellular function?
That's a good question. And really the answer is that, as the questioner asked, is to look at function. Typically, you would want to look at whatever type of an assay that you're using for the cells. What I have done is to use the cells in essentially a proliferation type assay, where I compare unstained cells, cells that have not been treated with any dye, with the cells that I've treated with the dye, and I can just count the number of cells. So it's just a count either on the Nova site using the counting function, or on a Coulter counter of sorts. And what you would expect is that if the dye has not affected the cells, that you would be getting equal numbers of cells in both of the assays. Of course, you can also do cytotoxicity assays and those types of things, but I think the proliferation, just the mere counting of the cells, is probably the easiest way for most people to do that.
Great, thank you. Well, that is all the time we have for questions. I want to thank all of you who have joined us today for this Biotechniques webinar on emerging instrumentation and application trends in flow cytometry. And we hope that the webinar has been informative and useful. I'd like to once again thank our 3 speakers for their presentations and ACEA for their sponsorship of this webinar. Goodbye.